So welcome back, everyone. We are here with Burcu Ayşen Hocam, and this session will be moderated by Selin Fahloğulları. So without going further, I will leave the floor to her. Selin, you may start.
Thank you, Ahmetcan. Hello, everyone. My name is Selin Fahloğulları. I'm a freshman medical student at Koç University and an active member of CUSAIN. Today, we will talk about pattern analysis and modeling method in cognitive neuroscience. But firstly, I would like to introduce our professor, Burcu Ayşen Ürgen. Burcu Ayşen is the assistant professor at the Department of Psychology and Interdisciplinary Neuroscience graduate programs at Bilkent University. She is also affiliated with ICEL's Supplementary Brain Research Center and National Magnetic Resonance Research Center. Ayşen received her PhD in cognitive science from University of California in 2015. After the PhD, she worked as a postdoctoral researcher at the Department of Neuroscience, University of Parma in Italy. She received her Bachelor of Science degree in Cognitive Science from Middle East Technical University. Currently, she directs Cognitive Computational Neuroscience Lab at Bilkent University. Hello, Jam. Thank you very much for accepting our invitation. It's a great pleasure to welcome you here. From here on out, I'll leave the floor to you.
All right. Thanks, Selin. Thank Ahmetcan. And thanks to the Koç University, Neuroscience Club for inviting me to this seminar series, to this festival, Neuroscience Festival. So it's kind of very nice. It's a pleasure to be here. So let me share my screen and then I can just get started. Can you see the screen?
Yes, Hocam, we can see.
Yes,
okay.
Hocam.
How about now? If I put it into view, you can see it. Okay, perfect. All right. So, okay. So today I'm going to talk about multivariate pattern analysis and computational modeling methods in cognitive neuroscience. I would like to make an introduction to the field, make an overview of what we know so far about the interdisciplinary work between pattern analysis, machine learning, from computer science and cognitive neuroscience. I will review some of the methods that have been used in the literature, and then I would like to complement it with some of our work in which we focus on perception of actions. All right, so let's get started. So I would like to first start with one of the most common methods that we use in cognitive neuroscience. I'm sure most of you have heard of it, functional magnetic resonance imaging, or the short name Fmri. So, of course, it is one of the techniques. It's not the only technique that we use, but to demonstrate how machine learning has revolutionized cognitive neuroscience, I think it's good to start with Fmri because it's the methodology where, the first, let's say, collaboration, first steps, in the collaboration between machine learning and cognitive neuroscience happened, right? So functional MRI or Fmri, it's a technique that measures brain activity using MRI technology by detecting changes in the blood flow, or known as also the BOLD response. So this method works on the fact that the blood flow and the neural activity are coupled. How does that work? It works in the following way. So when, let's say, one part of your brain is active, right? Let's say that there are a couple of neurons that are processing some kind of stimulus or some kind of, behavioral input, right? So there is neural activity, and then this neural activity requires oxygen, right? This oxygen need leads to increased blood flow to that region. And this increase in oxygenated hemoglobin affects MRI signal. And with Fmri, we measure the change in the magnetization between oxygen-rich and oxygen-poor blood. And I must say that it's not a direct measure of neural activity. It is rather an indirect, I would say, neural activity, because as I said, we are measuring blood flow to a region that is actively processing some type of stimulus. And it's a technique, nevertheless, it's a technique that's great to study human brain function, you know, so we can study live human brain with fMRI technology. And it is, another advantage is that it is non-invasive, meaning that it does not require surgery, it does not require drug injection or anything like that. So as long as you have, you know, participant that is eligible for MR scanning, there are, you know, several, you know, screening procedures that we have to do. You know, anybody can actually get an fMRI scan. And the nice thing is that it has a good spatial resolution, meaning that we can actually localize certain events to certain brain regions, right? So it's basically, it's a very nice technique that allows us to ask where certain things, where certain events are happening in the brain, but it has a bad temporal resolution, meaning that we can't capture the temporal sections of events very fast with the MRI technology. Okay, what we do with fMRI, you know, if you are, let's say, a cognitive neuroscience researcher, and if you are conducting an fMRI experiment, what you do is that you see this, this is an MR magnet, right? You lie in this magnet. It is movable, so this is a movable, let's say, bed. So the subject lies here, and then there's a head-mounted mirror here, and there is usually you know, screen behind the magnet, and the subjects see the stimulus, whatever you are showing, maybe images, you know, videos, words, whatever you are presenting, through that mirror, head-mounted mirror. The things on the screen through this head-mounted mirror. Okay, and then while subjects are doing this cognitive task, we take low resolution images, and after some, let me say, complicated data analysis techniques, we get these pretty brain pictures that basically show us which parts of the brain are active, right, are involved when you are actually performing a cognitive task. Okay. So in order to, I will walk you through what we do in order to get these pretty brain pictures. And then I'm going to tell you how actually we change the way we ask questions, and we now do not just ask which parts of the brain are active, but we ask different types of questions with pattern analysis methods. But before that, before I walk you through that step, I need to tell you what fMRI data looks like first, right? So fMRI data looks like a 3D, let's say, matrix. Those of you who may be familiar with, you know, mathematics, linear algebra, right? So it's basically a 3D matrix. And the basic unit of fMRI data is a voxel. So here you can think about this, your brain, and these are all, for example, 3 by 3 by 3 millimeter cubes. You know, these are voxels. And a brain image consists of thousands of voxels. And each of these voxels has an intensity value reflecting its activity in response to a stimulus, an input stimulus. Maybe when you show a face, when you show an image, when you show a house image, for example, or when you show a video or a word, whatever you are presenting, each of these voxels shows some kind of an activation. And I'm going to tell you how we compute that. And each voxel here, this is important, contains thousands of brain cells, meaning neurons. So we are not actually recording from a single neuron or anything like that. So here, you know, a single voxel can have thousands of voxels. Okay. And let's now look at a sample fMRI experiment, what kind of questions you can ask with traditional fMRI. So for example, you may have an experiment in which you would present people, your subjects, an auditory stimulus, maybe a song or some, you know, sounds, etc. And then you will have a rest condition where you do not present anything, just a blank screen. And what you are interested in is there a change in the ball response between listening and rest? So you might be interested in what parts of the brain, what regions of the brain are active, right, when you listen some kind of a sound, for example, and which parts of the brain are not active, right? And you compare listening versus rest. So this is a typical question that you can ask, you know, in traditional fMRI. And what you do is that you basically take these low resolution images, and here on the top figure, you see what we measure, the bold response. And here on the bottom, you see what we know from our experimental setting. So for example, the black line, the black parts here show our rest periods. So this is the rest period where the subject is shown, let's say, a blank, screen. And then these white periods are the periods where the subject is presented some kind of a sound. Okay. And then if you compare what is happening, you know, in a certain, let's say, voxel that we selected, let's say from auditory cortex, you see that the voxel is active when there is listening, you know, and then it's not really very active, right, when there is rest. So this is basically a voxel that shows this kind of pattern. And then in order to get those pretty brain pictures, right, like light up, right, what we do is in traditional fMRI analysis is mass univariate analysis. Here, basically what we do is that we build general linear models for each voxel to explain the data. How does that work? Let's say that we have our bold signal, right? And then we have our conditions in our experiment. So for example, maybe we have two conditions, listening versus rest. And what we do is that we basically specify our conditions, for example, listening and rest as regressors in a linear regression model, plus some error term, and we try to predict our bold response. And we do this general linear modeling at each voxel, okay? And then in the end, what we are trying to compute is basically, you know, we try to estimate these beta values that correspond to each of our conditions. And these would reflect the contribution of that particular condition. So for example, the contribution of listening or the contribution of the rest period to that particular voxel activity, the bold signal, okay? And then the next step in traditional fMRI analysis is that, let's say that you have a predefined, independently defined region of interest, let's say your auditory cortex, and let's say that you have 9 voxels within that region. And then what you can see is that you can look at how these voxels, you can check the voxel intensities during listening, and you can check how these voxel intensities change during rest. Let's say that these black ones show that these voxels are active in auditory cortex, you know, during listening, and only, let's say, 2 of these are active during rest. And by using what we call the subtraction method, you basically subtract the activity of these voxels, the activity of the rest period from the activity of listening voxel, and then you can identify the resulting activity pattern that corresponds to listening as opposed to rest, okay? But the problems, there are some problems with these mass univariate analysis that actually lead us to come up with some, you know, important machine learning techniques to be applied on a cognitive neuroscience data. So what are these problems? So first of all, the first problem is the fact that when you define, you know, region of interest, right? Let's say that instead of having a listening versus rest period, let's say that you are presenting your subjects 2 phonemes, right? They listen 2 phonemes. For example, one phoneme is ra and the other phoneme is la, okay? And then you basically look at these activity of these 9 voxels. And here, let's say that this is the voxel pattern that you see. This is the another one. So for example, here, 2nd, 4th, 6th, and 8 voxels are active. And here, the 3rd, you know, 6th, 8th, and whatever, 9th, 7th, and 9th voxels are. So different voxels are active. Some of them are common, some of them are not common. But in the end, what we do with traditional fMRI analysis is that we basically average over these voxels. So for example, when I average over these voxels, I may get an activation like this. And if I average over these voxels, I may get an activation of this. And the reason is that I have, let's say, 4 different voxels here, 4 different voxels here. And if you look at them, they are exactly the same when you take an average of them. right? So this basically shows the involvement of auditory cortex, when it processes raw phoneme or low phoneme. And if you compare them, they seem to be the same. They are not different from each other. But if you actually look at the pattern of responses here, you see that they are actually not identical. They are different from each other. Different voxels are involved in this. But if you take an average of these, you actually do not see a difference. So this is one of the problems of mass univariate analysis. It can discard very useful information that exists in the patterns of activity if you just use averaging technique. And the second problem is that mass univariate analysis assumes that each voxel is independent from each other. However, in reality, we cannot assume that the activity patterns in different brain regions are independent from each other, right? Because the brain is basically a network. So it's biologically more plausible to consider the patterns of activity rather than individual voxels, independently or separately. So here comes the use of multivariate pattern analysis methods, also known as MVPA methods with Fmri. And today I'm going to talk about 3 of these methods, that are commonly used in the literature. The first is the machine learning classifiers. The second is representational similarity analysis, and the third is voxel-based encoding modeling models. Okay. Let's start with the machine learning classifiers. So in machine learning classifiers, the basic question that you can ask is, can we predict a human's mental state by looking at their brain activity patterns? So you look at the brain activity pattern and you try to understand what the person is seeing, what the person is thinking, right? So it's basically a decoding approach, right? So, and one particular, let's say, question that you can ask is, let's say the subjects are seeing a face or a house. Okay, you present yours in an Fmri experiment, you present your subjects a face and a house, and then you record brain activity. And then without knowing what the subject is looking at, you look at the brain activity and you try to predict whether the subject has seen a face or the subject has seen a house. Okay, so in step 1, you present these stimuli to your subjects, you record your brain activity, which is basically, let's say that each of these squares show a voxel pattern, let's say activity, and this is basically the pattern that you get when you have a face or when you have a house. And importantly, remember, in fMRI experiments, you have to present your stimuli multiple times in order to get a robust signal, right? For example, when you have a face condition, you have to probably present it maybe 20 times, 30 times. And the same is true for house images. So when you present a house image, you can present it 20 times, 30 times, or like even more than that. So you basically have examples or trials for each of these stimulus categories. And then in step 2, what you do is that you basically vectorize these voxels. So you basically put together all of these voxels in a vector and you define them as a feature, right? A feature representation, a feature vector. And then you give each of these, feature representations a label. So for example, you have the voxels here, let's say from voxel 1 to voxel 100, right? You present them as a vector here, and then you give them a label. For example, this vector representation corresponds to a face activity, or another one is a house activity. And in the third step, you basically divide your data into training data and test data. In the training phase, you basically train a model and teach that model what a face activity looks like and what a house activity looks like. And then with your test data, you do not give the labels, but rather you give just the feature vectors. And then you ask your feature vectors, meaning, some kind of brain activity. And then you ask your model, you must have learned what a face activity looks like and what a house activity looks like. So predict, can you predict what this activity is? Is it a face or a house? And then if you have a good prediction accuracy, right? For example, if you have two categories, maybe above chance is 50%. If you get prediction accuracy above 50%, right, you can basically say that I can basically predict, you know, brain activity, what the person is seeing based on this, you know, patterns of voxel activity. But then this may not be very interesting for some neuroscientists, because they may say that, okay, I can decode, but what can I learn about this? You know, what can I learn with this about the brain? Can I learn anything about what kind of information processing the human brain maybe is doing, you know, using this kind of technique? And the answer is actually yes. And in order to demonstrate that, I would like to give an example from a very well studied domain, namely orientation selectivity in primary visual cortex in primates. So we know that from cell recordings in monkeys, for example, in macaque monkeys, the V1, the primary visual cortex, shows orientation selectivity. What does it mean? So, for example, if you show a bar like this, right, or let's say at 90 degrees, you see, so this is basically the action potential, you know, firing, you know, the firing of a neuron, right, when it is presented a bar like that, right? Or if, for example, it is presented something like this, it fires less. When it is presented like this, it fires less. When it is presented this orientation, it fires less. And if you, as a result of, you know, recording all of these activities, and then if you plot, you know, the response profiles, you actually get a tuning curve like that. So, and then there are definitely other, let's say, cells, neurons that are not sensitive to this, but that are sensitive to this, or that are sensitive to this. Okay, so for this particular, let's say, neuron, we can say that it is sensitive to, you know, this type of orientation. Okay, so this kind of knowledge is gathered from single cell neurophysiological recordings, right, in macaque monkeys or in other you know, non-human primates. But in humans, we didn't have any information about that. We didn't know whether human brain, human primary visual cortex has orientation selectivity. And because we can't do usually cell recordings in humans except special circumstances, right? For example, patients going under brain surgery. And then the question is, can we actually find a way to find, you know, to actually test whether human primary visual cortex is orientation selective? And can we do this with fMRI? Normally, if you ask, you know, let's say 2005, I would say, and anybody would say, no, you can't do that, because as I mentioned at the beginning of my talk, one voxel, which is the basic unit of fMRI, contains thousands of neurons. And then each of these neurons within the voxel may have a different orientation selectivity. Some of these neurons might be selective to, let's say, 90 degrees. Some of them might be selective to 45 degrees, which means that if you take the whole voxel, you might not get with fMRI, right, the orientation selectivity pattern, because it doesn't have that spatial resolution. But use of MVPA, multivariate pattern analysis methods with fMRI, actually has allowed us to identify orientation selectivity in human visual cortex in 2005. So how that thing happened, let me try to summarize it. So the idea is, can we reliably predict which of the 8 orientations the subject is seeing based on fMRI activity patterns in human visual cortex? So what the researchers have done is that they show these kind of grading patterns. For example, this is one pattern, let's say 45 degrees, and they show 8 different patterns like this that have different orientations. And then they basically record first brain activity from primary visual cortex. Then they basically train a decoder, a classifier, to actually learn the patterns of activity that correspond to each of these orientations. And then with their test data, they try to predict whether what the subject is, you know, seeing, what kind of orientation the subject is seeing, based on the Fmri data. And what they have found is that they find, above chance accuracy and very actually high accuracy, which means that they were able to predict what kind of orientation the subject is seeing by just looking at the Fmri activity patterns of primary visual cortex. So this was thought to be a very successful experiment because before this kind of analysis, if you do a traditional univariate analysis, you would not be able to see a difference between different orientations, okay? So by only considering the patterns of activity, you can get this information. And the interpretation of this finding was that maybe when you have a voxel, although it contains thousands of neurons, which may have different selectivities, each voxel might have a certain bias towards, towards a certain orientation selectivity, as a sum up of the, cells within that voxel, and that is reflected, that is captured by the machine learning classifier that aims to decode these different orientations, okay? So this was kind of quite a big success, and it just started the use of MVPA methods in cognitive neuroscience, especially with Fmri data. But of course, machine learning classifiers are not only used with Fmri data, they are also used with other imaging modalities, for example, like EEG, and one of the applied domains, brain-computer interfaces, people can actually use these classifiers to train EEG data and test EEG data, and then even control a device. And it can be a wheelchair, for example, or it can be, let's say, a robotic arm or robotic limb. So they can basically, with the brain activity patterns, you can actually, control these type of devices. So this is one applied domain, let me put it that way, but it is also applied in traditional cognitive neuroscience research with EEG, MEG, and even ECOG data as well. Okay. Let's now move on to the second pattern analysis technique, which is representational similarity analysis. So this is an analysis technique, or it's a general framework for testing computational and conceptual models. And it's a nice technique to quantitatively relate population codes between different brain regions, different individuals, different species, and different modalities in terms of their similarity. And in order to demonstrate how this method works, I'm going to give another example from object recognition domain. It's a very well-studied domain in cognitive neuroscience. Object recognition means, for example, distinguishing an object from other types of objects. For example, in order to say, you're presented all these different images of a car, and then you're presented all different images of other objects, and you're able to say, this is a car and this is not a car. This is a car, this is not a car. So being able to distinguish between a car and non-car object, this is object recognition, right? And we know that the inferotemporal cortex of the primate brain, responds to to objects, and actually, early research has shown that there are in inferotemporal cortex there are different patches that correspond to different categories of objects. So, for example, the green areas show selectivity for bodies, the red areas show selectivity for faces, some of the other areas show selectivity for houses or, scenes. And then there are, yellow areas that are selective to other objects. So early on, people have thought that there are all these category selective patches in the inferotemporal cortex. So there are all these modules. But later on, people have started questioning this thing because people have started to think that there might not be, so many patches in the IT cortex for each object category. Can there be more a distributed neural representation in IT cortex? And in order to test this hypothesis, Kirkes-Korte et al. Did a study in which they use a big set of object images, and then they recorded fMRI activity in humans, as well as single cell recordings in monkeys with the same set of stimuli. And then they apply this method called representational similarity analysis. And let me try to demonstrate how the method works. Let's say that you are shown a face or an image, like say house image, and then you record brain activity patterns. So here, all of these show your, let's say, voxel patterns. And then you construct what's called a dissimilarity matrix. Here, for each pair of stimuli, so for example, for faces and for houses, right, you have your voxel patterns, you compute their similarity, and this similarity can be basically Euclidean distance, correlation, and so there are many metrics that allow you to actually compute the similarity. And then in the rows and columns of this matrix, you basically enter the similarity of each stimuli type. Okay, stimulus type. So you basically show them. And then you basically measure how similar these images are. So for example, if you look at, so here, if it is reddish, right, it means that they are dissimilar. If they are bluish, it means that they are actually very similar. So in the case of, if you compare the similarity of a man face with a man face, the same, it is basically, you know, blue, the darker blue, right? But if you compare, let's say, a man face with a woman face, it's, let's say, a light blue. But if you compute a face, the similarity of a man face with a house, for example, it is yellow. It is this similarity increases, right? So it's basically a matrix that allows you how similar the representations of each stimuli are to each other. Okay. And then the next question you can ask is, let's say that you have this dissimilarity matrix. What can you do with a representational dissimilarity matrix? First of all, the first thing that you can do is representational content of specific brain regions, right? You can basically examine how a region is representing a certain type of, let's say, stimuli, right? How, for example, when Crickes-Corte, you know, applied representational similarity analysis on the inferotemporal cortex, which is known to be, you know, an important region for object recognition, they actually find this dichotomy between animate objects and inanimate objects, as you can see in the matrix representation. And then within the animate categories, they actually see this nice distinction between, for example, human versus non-human animate objects, and within the human objects, for example, bodies versus faces. So they start to see this kind of a pattern, right? And which basically shows that there are not only like patches, there is actually a distributed brain activity in IT cortex that correspond to many of these animate, inanimate, natural, artificial categories. The second thing you can do with representational similarity analysis is that you can examine how representations change along the cortical hierarchy, meaning that, so you can look at the, for example, how the dissimilarity matrix looks like in IT cortex, and you can compare it to another region. For example, early visual cortex. If you compare, for example, the dissimilarity matrix of early visual cortex, you see that there is no structure here. But, you know, from early visual cortex, when there is no categorical information, for example, between animate and inanimate objects, or faces or non-faces, are natural or artificial objects, you start to see these kind of, you know, distinctions between, you know, these categories in IT cortex. And the third thing that you can do with representational similarity analysis is that you can compare representations between different species and computational models. So for example, once you have your dissimilarity matrix, you can take the IT cortex from humans, you can take the IT cortex from monkeys, or you can take the IT cortex representation of a computational model, and then you can basically compare how similar they are, and you can try to infer many things based on these, you know, findings. And actually, Crickes-Corte et al., you know, in their study, they compared Fmri record dissimilarity matrix that is computed as a result of the Fmri recordings in humans, and then They also compare the dissimilarity matrix, right? That is found as a result, that is computed as a result of recording, from cells in monkeys, and you see how similar the representations are. So this is fMRI activity in humans. This is cell recordings in monkeys with the same type of stimulus, and you can see how similar the representations are. So basically, the similarity matrix or representational similarity analysis allows you to basically compare different modalities, different species, etc. So this is kind of one of the advantages of this technique. And then this is basically just an overview of what other things you can do. So for example, with human fMRI, let's say that you have a specific region, let's say your auditory cortex, and you can compare it with subject 1 and subject 2. So you can compare individuals. Or for example, you can have a subject, let's say subject 2 in these cases, and you can compare region A and region B. Or you can compare human fMRI, monkey fMRI, or you can compare monkey fMRI with monkey cell recordings, or computational models. So all of these techniques, all of these comparisons is possible, right, by having a dissimilarity matrix that is just a representation of how similar the stimuli are in terms of the neural activity patterns. Okay. I would like to check how much time I have. Okay, so the last technique that I'd like to talk about is the voxel-based encoding models. So here in the voxel-based encoding models, the idea is to build and fit some models that specify how information is encoded within a brain region and fit to data and evaluate the accuracy of these model predictions. So it's kind of a nice method because it basically provides you a complete functional description of a region of interest in the brain. So for example, you have let's say a certain brain region, let's say your V1, right? And you are interested in what type of computations V1 is doing. And then what you can do is that you can build several models. You can say that I have an hypothesis about what V1 is doing. Let me have this model, model 1. Then you can have another, let's say, hypothesis about what kind of computations V1 is doing, and you can have a model 2. And then once you have this complete description in terms of a computational model, you can basically test these models by fitting the models to your brain activity patterns, that you collected with fMRI. So let me give an overview of this method. So the first idea is that you collect data, right? For example, you show your stimuli, whatever, maybe in a vision experiment, for example, you show images or videos, right? And then you collect activity in the brain, maybe from certain brain regions or the whole brain. And then you divide your data into training data and testing data or validation data sets. And then you use your training data to estimate one or more encoding models for each voxel. Okay, so you have basically some future representations that correspond to, each of the categories that you have built in your model. And then you apply the estimated encoding models to the validation data and evaluate the prediction accuracy of the model. So you basically have your model predictions and you have fMRI data results, and you compare the model prediction with the fMRI data, and you see how good is your model in predicting the behavior, the fMRI data. And if there is good prediction accuracy, you can say that the brain might be using those features. The brain might be encoding that kind of information there. And here the important thing is that maybe using not only one model, but multiple models would give you more information, because if you have multiple models, you can compare the prediction accuracy of different models, and you can choose the model that is, let's say, that explains the data better, as a possible model, that shows the, information encoding in that region. And there's actually a study that used that is one of the original studies here, K et al., which basically uses an encoding model to predict the activity of primary visual cortex V1 in humans. They use grayscale natural images. They show people these, images like this, these grayscale images, and they built an encoding model that basically shows that basically encodes low-level stimulus features based on the known physiology in V1 and V2 neurons based on, physiology studies, as I said. So for example, we know that in primary visual cortex, as I said, there are all these orientation selectivities. They basically have these receptive field models where, so each voxel has a certain, let's say, bias, and they have certain activity, and they basically convolve it with some, important, let's say, aspects of fMRI data like HRF, etc. So I can tell you more about this if you have any questions. But in the end, they basically have this model, and then they estimate a model, they train a model, and then they basically try to, in the third step, they try to predict brain activity for a set of images using these receptive field models. For example, let's say that this is an image, this is recorded brain activity, and this is what you get, right? The voxel activity pattern. And then once you predict, once you, train, with the, once you use your training data, right, with the receptive field model, let's say that you get let's say, activity like this, right, based on your receptive field modeling, you compare them, right, and you select the activity whose predicted activity is most similar to the measured brain activity. Okay, so this is basically another method that allows you to understand what kind of information is encoded within a certain brain region. This was the first study, this K et al. And then following that, there are many studies that actually looked at other brain regions. So there are, for example, studies that look at the motion sensitive cortex, for example, MT region. There are other studies that look at some higher level visual regions, for example, FFA or PPA, some object selective cortices. And then for all of them, of course, you use a different method. And the idea is at different brain regions, for different brain regions, you need to define a different model because they would tell you what kind of information processing is being done in that particular region. So in that sense, it's kind of a very nice method that allows you to compare different models with some rigor, with some, precise, models. Okay, so finally, in the last part of my talk, I would like to talk about our work, our lab's work on visual perception of actions or action perception, in which we use one of these methodologies. But before I talk about that, I would like to just give you a brief information about what action perception is, what we study in general, what our research questions are. Okay, so the biggest question we ask is, how do we understand others' actions? We are interested in how we perceive, the people who are acting around us. Okay, how do we perceive the agents who are acting around us? How do we understand what kind of actions they are doing? How do we predict what they are doing, etc. So these are all the questions that we are interested. And as you can imagine, you can see people in a lecture. Most of you are probably in the medical school, in surgeries or in grocery shopping, at work, in traffic. You constantly see these people in action, and we are trying to understand their intentions. So our research question is to actually understand how we perceive these actions, how we understand them. And if you ask me what do we know, from neuroscience, regarding action perception, I can tell you that there are two, let's say, parallel lines of research. One line of research focuses on the visual system. And here the idea is that when you see an action, for example, someone who is running, there are two important visual cues. One is the form of the actor, which is basically the appearance, the body, for example. And the second cue is the motion, right? The movement pattern of the actor. And visual neuroscientists considered the following. They think that there are two parallel pathways. We know it very well since 1982 that there are two parallel pathways of the visual system: a ventral pathway, which primarily processes form information, and a dorsal pathway, which primarily processes motion information. And the idea in action perception is that when you perceive someone, who is acting, you basically use these two parallel pathways, in combination, and then at some point you integrate this information, probably at the level of posterior STS, to give the percept of a moving entity. Okay, and there's a second line of research that focus on the areas, brain regions that are beyond primary visual cortex or early brain, let's say, regions, and they focus on what we call the core action observation network or core action perception network. And this network consists of these three regions: posterior SCS, some regions in parietal cortex, and some regions in premotor cortex. So, cognitive neuroscience research so far have revealed these are the important regions that process actions, but there are some key questions that haven't been addressed. One type of question is, what does each region of the action processing network do? What kind of information does each region process, and how does the information change across the different regions? So these were some questions that haven't been answered. And this is why we conducted an Fmri study to address these questions. So in our Fmri study, we had a relatively, a large set of actions. We had 3 agents: a robot agent, and an android, a human-like robot agent, and a human agent. And then we had 8 different actions of these. So in total, we had 24 different movies. These are like 2-second short movies. And we presented them to human subjects, and we recorded brain activity. And with traditional univariate analysis, in the first step, we identify which brain regions are active or which brain regions are involved during action perception. And we found that, primary visual cortex, early visual cortex, extending to, posterior STS, parietal cortex, and premotor cortex are involved in action perception, consistent with previous literature. So this was basically a nice validation of previous work. So this was nice. But we were interested in beyond activation, beyond what is involved, right? We were interested in understanding what type of information is processed in STS, what type of information is processed in parietal or premotor. We were interested in that. So this is why we use representational similarity analysis. What we do is that we show each of these agents and their actions to our human subjects. We recorded brain activity. These are our activity patterns. And then we compute the similarity of brain's response patterns to agents and actions in each region that we are interested in. And in order to compute this, we use actually computational models. So one type of computational model is coming from computer vision. I'm not going to go into the details of these models. If there are questions, please ask me, at the end. So we basically use 5 different computer vision models that are commonly used in action recognition. And here we try to specify the form information in the movies, the motion information in the movies, the low-level form and motion information, and high-level form and motion information in the movies. And here our idea was that which brain region in the action observation network, might be processing this kind of visual information. Okay. So here we basically, based on all of these 9 different models, there are different variations of these models, but based on this, we basically built these representational dissimilarity matrices for each of these models, based on our stimuli. And then we also built categorical models by basically labeling our videos with certain features from our, stimuli. So for example, we label what kind of agent there is in the video. We label whether the motion is biological or not. We label whether the appearance is biological or not. We label whether the intention of the action is maybe manipulative, communicative, or it's a self-movement. We also, for example, label the target of the action. For example, the target when you perform the action, is it towards a human or towards an object? And we also label the action categories. So, and we were interested in this type of high-level information. Is it possible that this type of high-level information is encoded in some part of this action processing network? And these are the representational dissimilarity matrices for our categorical models. And in the end, what we did is, once we have our representational dissimilarity matrix for our brain activity and representational dissimilarity matrix for our computational model representations, we actually link them with correlation. Okay. And what we found is that when we look at the computer vision models and brain correlation, we found that the posterior STS, which is basically the closest part of the action observation network to visual cortex, it basically shows the highest, let's say, correlation with the computer vision models. And among the many computer vision models, we found that the models that contain the high-level form and motion information are correlated best with the STS activity. Whereas when we look at the categorical model representations, the high-level information in the movies, we found that parietal cortex is the one that is correlated best with these models, and especially the intention model, the target model, and the action model were the ones that correlated best with the parietal model, whereas premotor did not correlate neither with the categorical models nor with the computer vision models. So in summary, we can say that the three levels of the action observation network code for different aspects of actions. It seems that posterior STS codes for high-level movement kinematics, possibly integrating global form and motion information. Parietal node codes for high-level visual and semantic aspects such as action type, intention, and target. And premotor codes, neither of them possibly is involved in effector specification, which part of your body is, you are using when you are actually, processing actions, which could be probably tested in future studies, because in our movies, all actions were performed with a hand. And then action observation network seems to be a hierarchical system in which increasing levels of the cortex codes for increasing complex features. So I think this is the take-home message from this pattern analysis study. So in conclusion, I would like to say that machine learning and cognitive neuroscience, they have started to collaborate very, let's say, intensely in the last, 15 years or so. And machine learning has been very useful to build features and computational models of the brain. And it basically allows us to, build models and test these models. And then, when you have these models and when you have your neuroimaging data or cell recording data or any kind of data that you can gather from the brain, right, you can actually test those models, those features in your experiments. And then based on your results, you can actually improve these models, to actually account for your brain data much, much better. So in that sense, I think there is this, machine learning researchers and cognitive neuroscientists should work together, if we want to understand, human brain, and not only human brain, but in general, the brain, right, the nervous system very well. And I would like to start thanking my lab, who, allow me to, continue this line of work, after my PhD and postdoc years. Okay, thanks a lot. And now I can take any questions if there is.
Thank you so much, Hocam, for this comprehensive presentation. There are a couple of questions that our participation asked. I can start with one of them. One of our participants asked, does this modeling show any irregularities when it comes to the different patients? For example, is it any difference in autistic people or so on?
What is different? I'm not sure if I understand the question correctly. So the question is, can you use these methods in patient studies as well? Is it the question?
The question is about if we use this methods in some kind of different people, the results are same or not? Like if we use them on the autistic people, the result will be same or not?
Okay. So yeah, I think definitely these methods might be very useful if you want to compare, for example, different populations, definitely. So for example, if let's say that you have an hypothesis about, let's say, people who have autism spectrum disorder, let's say that they have problems in perceiving, let's say, actions, right? Because, these people may not feel comfortable in looking at people and how they, let's say, act, right? So let's say that you present some videos to these people, and then you present these videos to, let's say, healthy, humans as well. And then you compare their, let's say, activity patterns in the action observation network. So you can do traditional analysis, traditional Fmri analysis, and you can look at which brain regions are involved, active, but maybe when you compare people with autism spectrum disorder and people with healthy, let's say, humans, right? And then maybe you didn't find any difference. And then you would normally, before machine learning era, you would say, oh, there is no difference in brain activity, when you compare people with autism spectrum disorder and people, let's say, healthy individuals. But nowadays, with the advent of these machine learning methods in cognitive neuroscience, you can actually say you can't actually just stop there, right? You need to basically probably look at the patterns of activity. You can go and look at, for example, each region of the action observation network, or some, face processing network, or whatever network you are interested in, and you can do these pattern analysis methods separately, right, with people who are, let's say, autism spectrum disorder and healthy individuals, and then you can compare them. You can there are also techniques where you, for example, in order to understand quantitatively how they are different from each other. So, for example, you can train your data on healthy human data, and then test your model with, for example, autism people, and then you can see whether you can predict or not. So this is also something that people do. Definitely, I mean, for any kind of patient study, you can definitely use these methods. So these methods are not definitely restricted to any kind of, like only healthy humans or any other, let's say, methodology. It's commonly used in EEG research as well. So I only gave some, Fmri examples here, but it's definitely used in EEG research, MEG research, intracranial recordings, ECOG, and cell recordings, single cell recordings, multi-cell recordings. In all of these, areas, machine learning methods are being used. So they are very powerful, in understanding, let's say, previously unknown, aspects of the brain.
Thank you so much, Hocam. The other question is there a current or ongoing problem that MRI have that scientists are trying to solve? Like definitely. The method we use.
Yeah, definitely. So yeah, of course, I mean, none of this research, none of the methods that I told you, they are always progressing, right? There's never we never say it is the end, we can't do anything anymore. We can't say that, right? Because science is a progressive activity. It's basically an accumulation, right, of knowledge. And then meanwhile, in MRI methods, there are definitely let's say, methodological improvements that researchers are working on. So, for example, one thing here is that I told you, with Fmri, the spatial localization, yes, it's a very good spatial localization, but we say that we see, voxels that have thousands of neurons, and we can't actually, go beyond that. But actually now with high field MRI techniques, so one of the techniques, some of the techniques that I showed, the examples that I show here were the Fmri data that were collected at 3D MRI machines. But then, for example, we now have high field MRI machines like 70, even like 10T right now. So this means that as we increase the magnetic aspects of the MRI technique, you can actually get a better resolution. So you can actually look at a, let's say, higher resolution at the brain regions that you are looking at. And when you have that, you can basically use, again, the machine learning methods or even the traditional methods to actually look at what kind of information is being processed in these regions. And I mean, neuroscience research is really so rich. I think you have seen in the last 2 days, there are so many areas like language, right, consciousness, and memory, perception. So in all of these fields, these methods are applicable. So there are so many unknowns that we don't know, right, that we try to discover. So in that sense, from a methodological, from the empirical side, there are many questions that are not addressed yet. And from the methodological side, definitely there are researchers who are, I'm not the one who is working on, let's say, the MRI, let's say, technology, you know, in terms of improving the methods, but there are definitely researchers who work on how to improve the MRI technology so that we can get better images, we can get better signal from the images, etc.
Thanks so much, Hocam. For the last question, I picked something kind of related to your topic, but not today's seminar. One of our participants asked, which career route should we take as a computer engineering undergraduate student pursue in neuroscience research field?
Okay, so if I understand correctly, the person is doing computer science, computer engineering, BS, right? Undergraduate training. Okay, so if you want to do neuroscience, what should you do? Well, the answer is that I think, first of all, when you're in computer engineering, make sure that you take some important courses that would be very relevant for neuroscience. So, for example, make sure you take, you know, artificial intelligence, machine learning, computer vision. You know, these are very important courses, you know, or let's say, you know, if there is anything like that, Bayesian, for example, modeling. So these are very important, you know, subfields of computer science, you know, for neuroscience research. And then I think the second step is to actually go into a neuroscience program. And, you know, at my time, when I was a student, you know, in computer engineering, my background is also in computer engineering, there was no neuroscience program that I can get into, you know, as a graduate student. All neuroscience programs were under medical schools, and medical schools were not accepting people from different backgrounds. Like generally, they were accepting only, you know, students with medical degrees. So this is why I was not able to get into any neuroscience program at my time. And this is why I wanted to apply abroad, because, you know, there were schools definitely who were accepting people with mathematical backgrounds into neuroscience programs, into cognitive science programs. But things have changed in Turkey, I must say. I mean, I think Koç is definitely one of these universities where you have an interdisciplinary neuroscience program. At Bilkent, we have an interdisciplinary neuroscience program. I mean, we have like half of my lab is engineering, half of my lab is psychology, I must say. So it's basically now it's possible that, you know, with an engineering degree, you can get into, you know, in our, you know, country as well, there are definitely very, you know, nice neuroscience programs you can get into. And as I said, in our country, it is, you know, a little bit new now, but if you go abroad, you can see that, especially with the revolution machine learning has made in cognitive neuroscience, people with mathematical backgrounds are really wanted in cognitive neuroscience. Sure, and those of you who are in psychology, or those of you who are in, you know, other fields, should really like take, you know, really like computational courses if you want to go into, you know, neuroscience. I mean, we are really in an interdisciplinary era. There is no way that you can put boundaries between disciplines. If you want to understand the brain, you can't just say, I'm just going to use, you know, these type of methods, nothing else. Like, you would limit yourself and you would not see the big picture. So this is why I think you should be able to, you know, you know, push your comfort zone and then explore, you know, different fields, different methodologies, and, you know, teach yourself as much as possible, I think.
Thanks so much, Hocam. It was a great pleasure to welcome you here. Before we move on, I would like to take a picture of all of us, if it's okay to you.
Definitely.
So if anyone can open camera, it will be great, so we can take the picture.